Hello and welcome to the latest episode of Duchenne Science Live. I'm Sid.
And I'm Neil.
Today, prompted by a review in clinical epigenetics, we're looking at epigenetic modifications in muscle regeneration and in the progression of Duchenne. And we better start with explaining what epigenetics is all about. It's not about the genes themselves, but it's about what controls those genes. And so it's the great sort of master control board, and which genes are on. They're active. They're being actively read and translated, and ending up as proteins being made from them, and which genes are being, mothballed or not having proteins being made from at any one time. And in a particular cell, which is for a particular function. So, for example, a muscle cell. So that cell has got a very particular function, and it'll have a very particular profile of which genes need to be active or not active at any one time, and that will be peculiar to the muscle cell at that particular point in its cellular little life. So,
compared to say a brain cell, I imagine it's a totally different expression.
Yes, and all this on-off activation or upregulation, downregulation, different words to describe it, but which genes are being used and which are not, and that's the arena of epigenetics. It's all about the switches, and it's all about what pathways are saying. Well, use these genes right now because we need those products, or you better switch those off because we don't need those right now.
And I know people talk about epigenetics as almost environmental effects on yes. cells and on genes, but I guess it's important to say environment here does not mean just busy streets or grassy fields. It means the environment of the actual cell, doesn't it?
Yes. So yeah, what the cell is doing, what its job is. So that's why it's as you say, a brain cell is going to, or even within the brain, is just huge variety of different cell types, whatever. So epigenetics can be in place over sort of, it's all happening over the lifetime of the cell, and it might be that some of the ways in which the genes are switched off, for example, might be put in place early on in the cell's development, and then they'll never be used again because they're not relevant for the life and work of that cell.
Yeah.
And there's different ways that genes are turned on or turned off. And there'll be different enzymes, so that will be involved, and different signaling pathways and all sorts of different things. So potentially lots and lots of different targets for influencing those processes in terms of therapies.
And so we've thought of a way to show how epigenetics works using some theatre lighting techniques.
Yeah, that's right. So if you have the idea that you have your genes are a bit like all the lights you could possibly have lighting up your stage in a particular scene in the play, all right, and one scene will require very different lighting scheme or lighting profile to another scene. So you might have set a scene in the garden, they're terribly romantic, wonderful, and you might need to spotlight your two leads at the front of the stage. Okay, so you will set up your lighting desk to do exactly that. You will have the colors that you want, the effect that you want. You'll be highlighting the certain area of the stage, and imagine that that's what's going on with your genes. You've got the right genes are on, doing the right thing at the right time in the right place to make that scene, that cell work as it needs to. And that scene could go on forever, and there might be little tweaks in it when we need to maybe add the effect of rain in the corner there for a moment or something like that. But that controlling of the lighting is what makes the scene that scene. All right, and if you had the wrong lighting scheme for we've got a battle scene going and we've got soft romantic lighting, it's not going to work. It's not going. What's going on?
And so while we can't bring a theatre to life, we can do this on a much smaller scale with two different coloured lights. So for this demonstration, we've got two different colour light bulbs. We've got a red one on the right hand side and a blue one on the left. And what you can see in the middle, we end up with nice magenta, purpley coloured light where they mix. And you can imagine these two light bulbs as the genes inside a cell. So a cell, all our cells have the same set of genes, but they use them in different ways. And we can show that just by varying the brightness of some of the lights. So if I reduce the brightness of the red light, you can see the colour changes. If I put it back up, I can do the same with the blue light. I can reduce the level of blue light, and again the colour changes. It goes from kind of purple through to red. But the key thing here is that I'm not actually changing the gene itself. I'm just changing how much light it's producing, how much that gene is turned on.
You've got the same tools. You've got this. The cell has the same genes available to it as pretty much any other cell, but it's going to use them in its own personal, completely individual way to do what it needs to do.
It needs to do. Yeah,
exactly.
It's always the same coloured lights we're using, isn't it? The same.
Yeah, it's always the same. So you set up your lights, ready to go in the way that you want them, and then you can manipulate them to your to your will. So in Duchenne, we know that dystrophin, the protein encoded by the dystrophin gene, has a role in mechanically sizing the muscle cells, but it also has involved in signaling in muscle cells and in the satellite cells, the muscle stem cells, and those signaling pathways massively influence which genes are switched on and off in those muscle cells.
And so what we see in Duchenne is probably genes turned off when they should be turned on, and the opposite, so turned on when they should be turned off, and both of those have a massive effect on a cell because it can't control what it wants to do.
Yeah.
That causes the laying down of fatty cells instead of muscle cells. But actually, if you look inside those cells, you see very different changes, at the epigenetic level. So you can look at epigenetics. You can take the DNA out of a cell and look how it's been altered by the cell to control it. And actually, if you take DNA out of cells from somebody living with Duchenne, you can find very, very different, they're called markers of epigenetics. So you find different markers of epigenetics, and you find that the things that control those inside a cell are altered as well. And one of those changes is in something called HDAC, for histone deacetylase.
There are a number of different ways of influencing gene regulation, turning things on and off. And one of them is to do with the histones, the proteins that the genetic material DNA is wrapped around, and the addition of or removal of acetyl groups it's a big we know that that's a big signal, like use me, don't use me. So if there's lots of acetyl groups on the histones, then they are actively, the genetic material around there is going to be being actively used.
Yeah.
And so you've got an enzyme there, which isn't it's an ASE. It takes things off. So this HDAC enzyme cleaves off these acetyl groups. That's its job. Cleaves, chops them off, removes them. So you've removed the epigenetic marker that this is the bit of genetic material that you want to be using. So if you can stop that enzyme taking those acetyl groups off, then you can keep the genes in that bit wrapped around those histones active, going, like the read me, use me. So that's the cell would be using those. Because these epigenetic effects are mediated by the same sort of mechanisms in all sorts of different types of cell in the body. So as I mentioned, this histone acetylase and DNA methylation, all these things that can involved in switching on and off genes. If you have a drug that influences this in some way, like these inhibitors of the HDAC, then there might be sort of universal effects that might result in a lot of side effects. But although side effects have been seen in these drugs, it also seems to be also effective.
And there's a clinical trial in Duchenne. So there's drugs out there that can inhibit these HDACs. They're called HDAC inhibitors, another imaginative name. And And there's one called Chavinostat that's being tested in clinical trials by Ital Pharmaco. They've got a clinical trial, a phase 3 clinical trial ongoing at the moment, where they're looking at the effectiveness of that drug in people living with Duchenne. And so this is another area where if you can identify these changes, develop drugs against those changes, it's more ways to target the cellular changes you see in Duchenne, because, I think, well, we've said before on these shows that it's likely that a combination of several treatments might be more effective, and something like this, which can correct gene expression, might help in addition to those other therapies.
Yes. The muscle stem cells or satellite cells, we said we'd talk more about them because they're super, super important. Because stem cells of any sort retain the ability to divide and produce new cells. Because most cells, once they are in existence, they differentiate, that is, they become specialized to the job that they need to do. They grow and they do that job for the lifetime of the cell. The cells you need to create new cells are stem cells, which they divide to form new cells of a particular type, and they also divide to produce new ones themselves. So they're self-perpetuating as well, because you always need more stem cells to create the other type of cells you need. So within the muscle tissues, you've got these muscle stem cells, the satellite cells, whose job it is to produce new cells destined to become muscle cells, and also satellite cells of the future.
And so although muscle cells and satellite cells are really closely related, if you were to look at the DNA inside those cells and look at the epigenetic markers, which bits of the DNA are being used, it'll be totally different, right? Because they've got a very different job in the body.
Yes, that's again, so epigenetics becomes massively important here, because if epigenetics are affected, then it can't be the cell that it needs to be. And that's what we see in Duchenne with the muscle stem cells. And so trying to influence those, and obviously because the satellite cells are responsible for the generation of all the new cell types within the muscle, then getting them to function correctly would be a brilliant step forward.
Yeah, and actually there's the drugs that target epigenetics are becoming, I hesitate to say widely available, but I think it's an interesting area for lots and lots of conditions. So this might be one where you might be able to use drugs from other conditions to help in Duchenne. So I think it's
just about which pathways you need to influence in order to get the results that you want.
I guess it's all about the more of these changes you know about, the more potentially you can develop drugs for, and the more you can maybe improve the muscle environment in the future. So I hope you've enjoyed our whistle-stop tour through epigenetics and what they mean and why they're important in Duchenne. Join us next week for what we're doing next week.
I don't know. What are we doing next week? Anyone got any suggestions? Write in.
Yes, use the comments below to suggest papers, and we'll have a look at any papers you suggest. So yes, please give us ideas for future shows. We're trying to focus on the latest research, so if you see anything interesting, do let us know, and we'll try and cover that in future.